Hello friends, I am Dhavalkumar Surti, and in this specific video I will talk about investigating media fill failures. So my friends who are involved in the aseptic manufacturing, injectable manufacturing, sterile product manufacturing must be aware that media fill is an essential compliance part of the industry and we have to perform periodic media fills. And sometimes, I pray to God nobody has to face the media fill failure, but sometimes we had a media fill failure. And it is a very important investigation. If we have media fill failure, then it is very critical investigation, and it takes lots of effort, knowledge and experience to conclude a media fill failure investigation. So in this specific video I will share some of my thoughts how we can effectively investigate media fill failure. Hope it will be helpful, and if you like, you can subscribe to my channel. So I will, in this specific video, I will talk about the gross failure which you are observing. So what happens if you manufacture media, you filter it, and next day you observe that it is filled into the vessel itself, into the sterile holding vessel itself, the media has been filled. Or if you start filling and after certain time of filling, you observe that the media is already contaminated. So how we can overcome such a gross failures, so that it will not impact, or it will save my time, efforts and resources which are gone into the media fill. So with respect to the holding vessel or the sterile holding vessel where we are filtering the media and storing, the main reasons which leads to the failure, some of them are the gaskets. If the gaskets are not totally integral, it will pass into the leak test. But if we are not checking frequently the gasket, then by the time the gasket starts to rupture, and it may not provide the adequate leak proof sealing, and there are many gaskets into the system, so that we have gaskets for the view glasses, gasket for the joints, gasket into the process piping, gaskets into your nitrogen purging tubing, gaskets where your inlet is there. So everywhere we are using gaskets. So the gasket should be checked periodically. There should be a replacement frequency for the gasket, and before media you must ensure that all your gaskets are intact, integral, and there are no damages. That is one reason. Second, we are having so many connections onto the tank, so many connections. If clamp is not properly tightened, if there are so many blanks, which blinds we can call, which are not properly connected. View glass, see, we don't open the view glass. View glass is for viewing inside what's inside the tank, but we are not frequently opening the view glass. So that connection, that view glass connection may not be the integral every time. So you have to ensure all your connections within the vessel, in the vessels, so that they are intact, integral, and will not provide any leakage, will not lead to any leakages during the media fill. So this is one. Second thing is the inside surface or inner surface cleaning of the sterile vessels. So many times you observe that we stick some tapes, the autoclave tape, the sterilization tapes inside, and that should not be done. But sometimes some people, due to lack of awareness, they for the sterilization validation they use tapes either to stick biological indicators or the sensors. And what happens, these tapes may not be removed completely, and it is sometimes in such a location that it will not be easily visible. So before you go for a media fill, inspect your vessel thoroughly from inside, that there is no contamination, no glue, no tapes which are there. Thoroughly clean them, so that one will say we are doing SIP. Yes, we are doing the SIP, but this contamination will provide a micro crevices, which is micron size crevices, which will protect microorganisms during the SIP process. They can provide sheath or covering, so that your SIP cycle may not kill those microorganisms and can lead to growth during your media fill and can lead to gross failure. So you have to thoroughly inspect your vessel from inside before you go for media fill, and this process should be for the routine also. So we should not be very cautious during the media fill, and we should not be very relaxed during our commercial production. Commercial production is our business, and we have to be very focused. Same attention during the media fill, and same attention should be provided to the commercial production also. So inspect the vessel. There should be frequent checking. You can maybe 3 months, maybe 6 months, you can inspect all your vessel from inside. Your cleaning program should ensure that there is a daily check or check after every cycle, every cleaning cycle, so that there is no contamination, no rough surfaces, no glue within the tank, which can lead to either media fill failure or product contamination. That is the second reason. Third reason is for the gross failure is your assembling itself. So we know for the sterile manufacturing we have to put filling assembly which will fill the product into the vial or ampules or pre-filled syringe, whatever you are filling line is for that purpose. So if you are not doing the assembling proper, it starts from the sterilization itself. If your assembly is not properly wrapped, not properly sterilized into the autoclave, that is one. Secondly, if you transfer your assembly after unloading from the autoclave, you transfer through mobile laminar air flow unit to your filling machine. If during that transfer, if it is not kept continuously under grade A, then what will happen? It can have contamination on the upper surface of the wrapping, and ultimately it can land into the your final filling assembly needles or tubings and can contaminate media. So your sterilization, your wrapping, your transfer should be very thorough, should be validated to smoke studies also, so that you can be sure that there is continuous laminar air flow or grade A air provided to the your filling assembly. And while you are assembling the thing, it should be validated process, it should be thoroughly verified through smoke studies, so that this entire process is not creating a aseptic intervention which can lead to your contamination into your filling assembly. So these are the three important points. One is your connection, aseptic connection, gaskets, view glass, everything we have to check. Second point is your inner surface of the sterile holding vessel, and third point is your assembling process, assembling, sterilization, wrapping and assembling process. It should be very thorough, validated through smoke studies, verified, and then only you should go for media fill. If you are not sure of this thing, you should first be sure and then only go for media fill. Because once it is a media fill failure, then it will be a very long investigation, and we have to put your manpower, resources, and most importantly time. You will delay your activities to investigate media fill failure and again to repeat it. So hope this will be helpful to you. Thank you for watching my video. Hi, I am Dhavalkumar Surti, and in this specific video we will understand about the media fill failure investigation. In my previous video we discussed about the gross failure of media fill, and in this specific video we will discuss about the failures which are not gross. For example, we are getting 2, 3 or 4 vials intermittently having a contamination during the entire course of media fill. So we will understand how we can investigate this intermittent was few of the contaminated units during the media fill. So first and foremost important part of investigating this single or 1 or 2 contaminated units is to understand at exactly what time the failure was occurred. For that we have to track the entire media fill activity. So right from the beginning of the assembling activity throughout the filling, and what were the interventions, how we filled, what were the interventions, how many people were involved, everything we have to, I think, tabularize that during this time this activity was there, filling was started at, assembling was started at this time, filling was started at this time, the number of interventions, the duration, time, the persons who were involved, everything we have to put in a sort of a table which will give a clear idea that these were the activity throughout the course of media fill. Now we have to correlate it with the failure contaminations. For example, we have 100 trays of media fill. Out of 100 we have failure in 10th, 15th, maybe 38th, 46th tray. In order to understand what was happened during that exact tray while media fill, we have to see when those trays were filled, what was the activity. That is the simplest way to correlate activity against the failure. If there is certain intervention, certain new intervention, certain unusual practice which was observed during this time, then you can correlate it. And for that it is most important that we should record each and every intervention, each and every activity during the media fill. Video recording is also very helpful for the purpose of investigation. It's not a mandatory regulatory requirement to have a video recording, but video record helps to investigate, helps to train people. So this is one part. If you are not able to correlate any activity with the failure at specific time point, then we have to go for the other things, like what was the environmental condition, what was the air velocity, what was the HEPA filter integrity, are there any failure with the HEPA filter, are there any concern, are there any cleaning concerns, was the maintenance of the machine was done properly, is there anything something within the system, is the assembling was not proper, which might not have done throughout the harm throughout the bed, but at some point it could have contributed. So we have to go in the depth of the things. If the obvious correlatable cause is not identified, then we have to go into the deep. We should check cleaning, sanitization, sterilization, SIP. SIP failure will mostly lead to the gross failure, but still we need to check what are there any new operators, or they have done any new intervention that we have to see. Have we done anything unusual that we have to see? Apart from this, while capping, or while the how the if it is a vial, how we have stoppered, if it is a PFS, how was there any unusual abnormal observation during the fill. So everything, this thing we have to consider while we are investigating the unusual few of the counts which are not throughout the batch but at some time point. So this is the way we can understand, we can go in deep and investigate how we can find a root cause about the failure. So hope this will be helpful. I will be making more video. If you like, you can subscribe to my channel. Thank you for watching my video. Hello and welcome to all my knowledge partners. Welcome to my YouTube channel PHARMAVEN. With respect to the overwhelming response on my media field failure investigation series on the videos, I am making this video to further explain how to investigate media field failures. So in this specific video, I will be talking about the media field failures which are associated with the complex manufacturing system. Complex system, I mean to say, where there are SIP skids, CIP skids, lot of vessels which are involved into the manufacturing system. So I will be talking on that part of media field. So friends, when we have skids into the system, it is good to have skids into the systems because it prevents aseptic connections. But the challenge with respect to the skid are the same as with respect to the manual connection. So when you have lot of skid, lot of vessel inside your manufacturing system, your filtration system, there are a lot of gaskets, lot of clamps which are coming into the picture, and you have to ensure that all the connections, all the clamps, all the gaskets are intact and properly sealed. How to ensure that? So in this specific video, I will be talking 3 important points. One is proper connections. 2, ensuring that each and every gasket, each and every connection is ensured for tightness before you proceed for the media field, and 3rd is leak test. So these are 3 important aspects when we are performing media field for a complex manufacturing system which involves multiple number of vessels. So let's begin. So when we are going for a complex manufacturing system and media field for the complex manufacturing system, you have to first ensure that your sterilization is robust. When I say robust, it has to be validated with considering all the requirement, all the connection, all the worst case scenario where you think based on your assessment that steam penetration or etching the temperature cannot be there, or we have to ensure that there are possibility of not achieving the temperature within this specific area. For example, if you have a skid of 2 to 3 filtration vessels, I will say, then the farthest end of this series can have the least exposure to the steam. So what you have to do, you have to place sensor in all your vessels, in all your critical connections, all your connections, and to the drain point of the last vessel in your series of filtration. So this is first first point of assurance that we have to ensure that your SIP is properly validated, so that each and every vessel in your filtration skid and each and every connection till the drain point is achieving the desired sterilization temperature. If it is not properly ensured, not properly assessed, then in your media fill you can get a gross failure in terms of it will be filling into the vessel itself. You don't have to go to the filling step. So that is first part. So ensure that you assessed all your placement of thermocouples within the SIP skid for the filtration vessels in the complex manufacturing system are properly evaluated, and we have ensured that either both temperature and biological indicator are meeting the acceptance criteria for that specific location. Once till that assurance is not done, please don't proceed for any further processing including media fill. Once you have robust SIP validation, then you plan for your water trial, at least one water trial. What mistakes majority of people are doing is that they are directly going for the media fill before taking a water trial of the entire system. So you have to take a trial, I'll say water trial, your placebo trial, that your manufacturing, your filtration, and your filling is working properly throughout the system. Once it is ensured, then only you go for the next step. Why? Because the complexity or the issues which may arise during your routine process, if you have not done the water trials, if you have not done the placebo trials with the product, then or simulating the product, then there are chances that you will find surprises during the media fill, which can lead to media fill failure. So this surprises, if arise during the media fill failure, then it will be a black spot in your system that you had a media fill failure. So to avoid that, please conduct a water trial with the system. Once you have done, you are sure that your water trial is successful, there are no leakages. See, one of the important aspect of your this skid system, where you are multiple vessels and multiple collections within the system involved, then leak test of the entire system is very, very important. You have to ensure that you are performing leak test just before your sterilization cycle, not after that, not after any intervention, and no any further connection or intervention should happen once you have done your complete leak test. Then you should go for the directly for the processing, which can be media fill, which can be product. So ensure that the entire your system is leak proof. When it is leak proof, then you go for your actual processing. So while going for the media fill, ensure that you have proper connections, you have proper gaskets, all the connections are ensured, there are no dead legs, no areas where the proper steam penetration is not there, including your filters, including your connections, including your vent filters, including your connection to the filling line, including all of your system where your steam is getting drained out. Ensure that each and every part of your system is getting required temperature. Once it is done, then only you go for the media fill. This is the one way of avoiding the media fill failure into the complex system where we have multiple vessels and sterilization in place technology, SIP in sterilization in place, which is called SIP, is involved. Then only you go for the media fill. By this proper, yes, of course, it will take some time. It can take a week to evaluate all these things, but it is better to spend a week on preparation rather than spending 2 months in your investing your media fill failure. So hope this helped. Thank you very much for watching my video. If you like, you can subscribe to my channel PHARMAVEN. Thank you to all my knowledge partners. Hello and welcome to all my knowledge partners. Welcome to my YouTube channel PHARMAVEN. In my series of video about media fill failure investigation, in this specific video we will talk about how to investigate a media fill failure when you have a complex manufacturing system. So in my one of previous video I explained that how to prevent media fill failure in the complex manufacturing system. But suppose anyhow it has been failed, now what to do? How to properly investigate the media fill failure? So first and foremost, Before moving forward, I request if you are liking my videos, please subscribe to my channel and watch my entire video, because it helps to reach more of the people. If you watch my full video, it will help the channel itself to reach to more of the people, and it will benefit more for all our pharma professionals. So my purpose is to share my knowledge and to make it available for as many people as possible in the pharmaceutical industry. So thank you. Please subscribe, please share my videos to all your friends, all your groups, so we can help each other to make a better pharmaceutical industry. So coming to the media field investigation part, when you have complex system, involves multiple CIP, SIP, skids, multiple filling operations, multiple connections, then if it is fails as a media field, then how to investigate? So first and foremost, when you identify that you have media field failure, all the technical skilled persons who knows the process should go to the area. That is the first and foremost requirement. Just go to the first hint or first intimation from the production floor that there is a contamination either into the vessel, either into the container. In the media field, during the media field, all the persons with technical skills should go to the area and take a overview of entire system, what is happening. Ask the people what you have done, and whatever you have done, is it part of our procedure, is it mentioned clearly in the system. If you don't go immediately to the area, then lot of information can be lost. People may, the operator who are conducting the media field can forget. So just go to the area and ask questions. Have we done anything different? Was there any abnormality? Was there any connection which was not done properly? What do you feel could have led to this failure? So these 3-4 simple questions can give you the correct answers. So first go to the area, analyze the entire process for yourself, check each and every connections, each and every gasket, each and every parameter, especially whether we have proper sterilization cycle done, whether we have done all the steps as per the SOP or the batch manufacturing record. Many times what happens, people see media field is a lengthy process and it takes 2 to 3 days. So there are high probabilities that people may skip or forget or hurry up with some of the steps which are critical to the process. And if that these steps are not done properly, then it can lead to your introduction of the contamination. For example, connections. So if you have complex manufacturing system, then there are a lot of connections which are to be done before sterilization as well as after sterilization. We have connectors which are available, which is called S2S connector. Once you connect after sterilization, it ensures sterility. But there are high probability that connection is done proper, but the remaining of the connection, if are not done properly, then can lead to introduction of the microbial contamination. Whether you have grade A by B or grade A by C, or you have isolators, it can happen in any of the area. And I have seen failures in all this manufacturing processes, all the type of technologies, whether you call it LF, whether you call it wraps, or whether you call it isolators. I have seen the failures in all the sort of areas because the connections are not ensured properly, are not done properly or defined properly into the SOP or into the batch manufacturing record. So ask the people whether they have done all the corrections correctly. If the answer is yes, whether we have passing the leak test of the entire system, answer is yes, then ask more questions after the leak test. Have you done anything, or have you disturbed any connection? Have you made any connection after your leak test passing or after your SIP? Have you modified anything? Have you checked or corrected anything which you feel is not good after the SIP or after leak test, and you have done? So this sort of information on first hand or during the immediate investigation will help you to understand that what might have gone wrong during your media field leading to the failure. So that is the first first and foremost requirement, to have a first hand information of your process. Once you have done it and if you find the abnormality, well and good, that you have the root cause. Yes, this connection was not proper, or this sensor was not properly placed, or this gasket was not properly kept, which might have introduced the contamination to the system leading to media field failure. But if you are not finding, then list out all the information, check with the process, check with the procedure. So whether we have clearly defined that what is to be done to ensure that it maintains the sterility for the system is written into the either into the procedure or into the batch manufacturing record of, or media field batch record or media field protocol. If the clear cut instructions are not written step by step, how to connect gasket, how to place gasket, how to connect a trichlor clamp, how to place a sensor, or how to tighten the gasket, or how to make one-to-one connection, how to make the aseptic connections. If the steps are not properly written, then there are a lot of chances that people by their own skill, by their own experience will be doing that activity. So one person will be doing correct, other person may not be doing the same activity in the correct fashion. So if there are such gaps, identify those gaps and fill in, or make it as a part of your either of your procedure or a media field protocol or a media field BM or batch record. So by that way you can prevent the reoccurrence. Third part is understanding about aseptic manufacturing. If you are person who are working into the area, if they are not acquainted with the aseptic manufacturing, with the how the grade C works, what is the grade C, what is grade B, what is grade A, what is isolator, if they are not aware clearly how it plays an important role in sterility assurance, if it is not clear, then there are a lot of chances that they'll understand whatever they are doing right and they do that activity which can lead to introduction of contamination to your system. So knowledge of your people is very, very important about your systems, about aseptic manufacturing, about aseptic connection, irrespective of you are operating with grade A, grade B, wraps, open wraps, closed wraps or isolators. If the people or the persons who are doing that activity, if they are not aware what is the importance of aseptic practices, what is important of connection, what is important of sterilization, then it can lead to the media field failure, and it is frequently occurring or frequently observed scenario when we investigate media field failure. So hope this helps you to understand more about media field failure investigation. Hope this helps. If you like, you please share to your friends and let me help spread the knowledge. Thank you very much.